Environmental enteric dysfunction, EED, is a disease of the small intestine that is thought to be a risk factor for growth stunting, a condition that affects over 150 million children. EED leads to disruption of the intestinal barrier and loss of absorptive surface area. However, since most children in low-income settings do not have access to endoscopy and there are a lack of validated non-invasive biomarkers, the etiology and prevalence of this condition is largely unknown. The Bangladesh EED study, BEAD, was designed to identify biomarkers of this condition and assess the relationship between the small intestinal microbiota and stunting in 12 to 18-month-old undernourished children. In this study, children whose stunting was refractory to a nutritional intervention were eligible for endoscopy with collection of duodenal biopsies and aspirates. Using culture-independent methods and aptamer-based proteomics, we found that the abundances of several duodenal bacteria from children with EED were positively correlated with biomarkers of intestinal inflammation and negatively correlated with linear growth. Finally, to assess causality, we colonized germ-free mice with a consortium of duodenal bacterial strains obtained from stunted children from the BEAD study. Compared to control mice, those that were colonized with the BEAD consortium developed an enteropathy similar to that of the children with biopsy-confirmed EED. Understanding the mechanisms by which members of the small intestinal microbiota affect mediators of linear growth will facilitate the development of new therapeutic approaches for stunting, including those that are designed to reconfigure the small intestinal microbiome towards a healthy state.